Hi everybody. Today we're going to move on to another learning target, one that you probably do have some background in already. Basically today we're going to cover controlled experimenting. So our goal is that you're going to be able to develop and evaluate a valid scientific experiment and be able to tell the difference between all the variables. In a controlled experiment, variables, the factors that might influence the results of an experiment, are recognized so that only one thing is changed and tested at a time. If more than one variable is changed at a time, then there's no way to determine what made the difference. So basically, in a controlled experiment, you're systematically testing one thing at a time to figure out how much of a difference it makes. Now, as you go through the video, please make sure you have a highlighter and please add anything that I have in yellow. Now, you can add as much as you want. You can watch this more than once if you need to. I do tend to talk quickly, so if you find that you're having trouble, you can stop the video and kind of rewind. Basically, in a controlled experiment, we always start with the hypothesis. But before we get to that, I do want to give you just an example of a scenario where we're going to pick it apart and be able to recognize all the variables. So my sample experiment, if you would add it to your sheet, is I'm going to talk about if I wanted to do a controlled experiment to determine how much of a difference talking to the plants affects their growth. So on your sheet, please add talking to plants can help them grow better. So this is the idea that I'm going to use to kind of help evaluate all the variables I'd have to keep in mind. Now, any good controlled experiment always begins with a hypothesis. Scientists don't just test things to test things. They're actually testing out things to either prove or gather evidence to support an idea that they have. So a hypothesis, you should be probably fairly comfortable with it. Please add a hypothesis is an educated guess used to lead an investigation. Data is collected to determine if the hypothesis can be backed by evidence. So in my example of talking to plants, let's say I have a hypothesis. My educated guess is that talking to plants could allow them to grow better since they get more carbon dioxide. So in a hypothesis, I have some background information. So in this example, I know, for example, that plants need carbon dioxide to grow. They take in carbon dioxide, sunlight, and water, and from that a plant can then make sugar and grow. So since I'm exhaling carbon dioxide as I talk to my plant, I'm going to hypothesize that talking to them could possibly increase their growth since I'm increasing one of the nutrients they need to grow. Now, in a controlled experiment, you need an investigation purpose question. The purpose question states what will be changed and tested. So very simply, it's usually just one question. So in this scenario, my investigation purpose question would be, does talking to plants improve their growth? So add that to your sheet. Now in this particular unit, you're going to do two different controlled experiments, and you're going to fine tune your purpose question into a one sentence question, such as mine. Now, this is the part that's slightly more complicated. Every controlled experiment has what's called an independent and a dependent variable. There's one word I'd like you to associate with independent. For example, your parents want you to be independent. Independent means on your own or one. So when you think of independent, I want you to think of one. Being independent means you can be one, you can rely on yourself. So specifically, if you would add on your note sheet, an independent variable is the one variable that is changed and tested in the experiment. And keep in mind that's the definition of a controlled experiment. If I'm changing more than one thing at a time, I really can't tell what made the difference. In my example, my independent variable, the one thing that I'm changing and testing, is talking to the plants. So talking to the plants is the thing that I'm changing and testing. So it's my independent variable, that only one thing that I can mess with at a time when I do a test. Now dependent variable, dependent, I also want you to have a one-word association. Dependent is basically measurements that are monitored. So I want to see how much difference does talking to a plant make. So my dependent variable, its definition is simply the measurements or observations collected during the experiment to see how it responds to what is changed and tested. So the dependent variable is what I'm measuring and monitoring to see if what I changed made a difference. And if it made a difference, was it a big difference? Was it a little difference? So in my example of I'm going to talk to plants to help them grow, what would I need to measure and monitor to see if talking to them made a difference? Well, if you're thinking the measurement of the height of the plant, you're probably on the right track. So I need to have some kind of data about how the plant's growing, either the mass of the plant or maybe even just simply I'm going to measure the height of the plant. So I have to pick one thing that I'm going to measure and monitor as I test my plants. Now, one of the things about a controlled experiment is you can't just do one test on one plant and call it done. That's not scientific evidence. Scientific evidence is repeatable. That means I can test a large quantity of things and I'm going to always get the same result. So constants, when something stays constant, it stays the same. So again, a one-word association for constants are all those other variables that have to be kept the same so that you're ensuring that only one thing was changed and tested in your experiment. So in the definition of constants, there are other variables that are kept the same throughout the experiment to ensure that only one thing is changed and tested at a time. If you're not aware of all the variables, this is where your controlled experiment might not work because you're not recognizing everything that could be influencing your result. So in my group of plants that I'm talking to, some things that I'd need to keep the same in order to have a fair comparison is number one is I probably want to have all the plants be the same kind. Secondly, I'm probably going to make sure they have the same growing conditions. They have the same amount of light. They have the same amount of water. So the only one thing that that a set of plants has different is that some are going to be talked to. So constants are all those growing conditions I have to keep in mind that stay the same so that a plant doesn't inadvertently do better or worse than another plant because of a different variable besides talking. Now, one last word, vocabulary, is a control. Control and constants, students tend to kind of confuse. So again, constants, think of things that have to stay the same. A control, on the other hand, if I had a couple of small words, is basically it's a comparison. It's a normal result. So some experiments have a control that is a normal setup, and a control demonstrates a typical result when nothing is being changed in the experiment. So the control again is what you come back to compare. If I did see a difference in how much the plants are growing as I talk to them, I could use the control to determine how much difference did it make. And again, a control is just simply a test where I'm just running it as normal under normal conditions. So in my example of talking to plants as a controlled experiment, I would need a normal plant, or more than one most likely, to compare against which will not be talked to. So I need to have a plant grow normally without being talked to so I can determine how much does it typically grow in, for example, the month's time that I'm testing out my talking to plants variable. Now lastly, any good controlled experiment always ends with the conclusion. Very simply, it refers back to the purpose question and it identifies the relationship found. Is there a relationship between what you tested and what you measured and monitored? And if there is a relationship, how much of a difference? Is there a direct relationship? Like if I increase one thing, it's going to increase. If I increase talking to plants, it's going to increase their growth. Or is it more like an indirect relationship where, let's say, for some goofy reason, I talk to plants, the more I talk to plants, the less they grow. That would be where I would have an inverse relationship where one variable goes up, the other one goes down. Now keep in mind, some experiments you may very well have no difference, where what you changed and tested did not make much of a difference, where you would say, I don't have any evidence to suggest that what I tested actually made much difference. So very in a nutshell, in my example, I would simply ask myself at the end of my experiment, did the plant's height change or stay the same compared to the control? Now, like I said, if you feel like you need to go back and add some more detail, please pause the video. Otherwise, let's head to the back side of your note sheet. On the back side, when we graph data, there is a very specific place where we put our variables. So I would like you to highlight the dependent variable. What is measured or monitored in the experiment always goes on the y-axis. That way you can kind of see, is one thing increasing, is it staying the same, is it decreasing? So in my example of the plants, I would be putting the plant height on the y-axis, because that's what I'm looking to see if it's changing. Is plant height staying the same? Is it increasing over time, or is it decreasing? Now the independent variable, now in this example I wouldn't put talking to plants on the x-axis. Many times the x-axis is simply like the time increment, where for example, I might monitor the height of the plants once a week. So I'd have week 1, week 2, week 3, week 4. And basically, I just need you to keep in mind the one variable that you have to know when you put together a graph is what you're measuring and monitoring in the experiment always goes on the y. Now, lastly, let's go through some guided practice where we go through a couple small scenarios and we need to identify our variables, the dependent, the independent, the constants, and the control. So in the very first example, the height of bean plants are recorded daily for 2 weeks when a fertilizer is used. So in this example, the dependent variable, what I'm going to measure and monitor throughout the experiment would simply be the height of the bean plants. So please add that to your sheet. Now if you feel like you need to reinforce how do I know that, then add what's in purple. So basically the dependent, again, is always what you're measuring and monitoring in the experiment. So if I'm going to be fertilizing bean plants for 2 weeks, I'm going to be watching their height to see if it's changing. Now the independent variable, remember independent means one, being on your own. What's the one thing that I'm changing and testing in this experiment? Basically the fertilizer. I'm going to use fertilizer on some plants and I want to see if that makes a difference in their growth. So again, please add using fertilizer is the independent variable. It's the one thing I'm changing in this particular scenario. Now constants again are the same things that you need to keep the same. So what will I need to keep the same in order to compare all the plants that I'm growing? Very simply, I'm going to need to, oops, sorry, wrong one. I'm going to need to have the same kind of bean plants, bean seeds, the same growing conditions, and these are the variables that need to be the same for there to be a fair comparison among all the plants, and to ensure basically the only thing that's different in my set of plants is that some get fertilizer and some don't. So my constants are the growing conditions, keeping everything like food and water and sunlight the same. Now the control are my normal result. The bean plants growing without fertilizer is my control. It's a normal result to see how much the bean plants growth without fertilizer, and it can determine how much difference did the fertilizer make. Did the plants grow twice as big or half it or 3 times as big as the ones without fertilizer? So again, a control is what I have as a normal result. Now let's go on to the second example. Hamsters are kept at different temperatures for 6 weeks. Body fat percentage is recorded. So in this particular experiment, I'm going to keep track of some hamsters, and I want to see if keeping them in a hotter or colder environment will affect their weight. So the dependent variable, what I'm measuring and monitoring throughout the experiment would simply be the body fat measurement. I'm going to keep track of my hamster's weight, their body fat. So again, the dependent is what I measure and monitor. The independent variable, what am I changing and testing with the hamsters? If you said the different temperatures, you're exactly right. So I'm going to keep them at different temperatures to see if that influences their body fat. So the one thing I'm changing and testing is keeping them in different temperatures. Now, if I'm going to have a whole bunch of hamsters, I need to have some constants. I need to keep some variables the same so that only the temperature that they're sitting in is different. So constants that would need to be the same are, if I have 20 hamsters I'm testing, I'd need the same kind of hamster. I'd need probably about the same age of hamster so that I have a fair comparison of hamsters. The same feeding schedule, the same amount of exercise. So these are all the variables that possibly would influence a hamster's body fat. And again, I only want to change one thing, which is the temperature at which the hamsters are kept. Now lastly, I need a control group for the hamsters. So if I want to see if keeping hamsters at different temperatures affects their body fat, I'm going to simply need a group of hamsters that are under normal room temperature conditions. So again, a control is a normal condition. It's a normal result so that I can see if the hamsters in an excessively hot environment have more or less body fat than hamsters that are just normally kept at room temperature. Now, last but not least, this third one, weight is monitored weekly to determine how well a diet pill works. So in this experiment, I'm looking at what am I changing, what will I measure and monitor throughout the experiment. I'm going to keep track of weight. Weight will be monitored throughout the experiment. Now, what am I changing in testing to see if weight is affected? That, of course, would be taking the diet pill. So taking the diet pill is the one thing I'm going to change and test. Now, what are some constants that I'd want to keep the same? So if I have a group of people taking diet pills, what would I want to keep in mind that is the same about all those people so that only the diet pill is going to influence the result? So constants would be, for example, I might want to have the same sex of people. So I might want to have all females in my study taking the pills. I might also want to make sure that they're generally following the same diet and have the same amount of exercise so that I don't have a group who's working out a lot and some who are and aren't working out, because obviously that would influence how much they weigh. So I have to be very careful in this experiment that the people that I choose are about the same age, the same sex, they have about the same diet and the same amount of typical exercise in a day. Now, the control group, I need a group of people to compare against so that I can see how much of a difference taking the diet pill made. So in my control group, I simply have people who are not taking the diet pill. So I could make a comparison and say, compared to the people who are taking the diet pill, this is how much weight they lost compared to the people who did. Now, lastly, on your note sheet, you should have one last example. I'd like you to go through that example on your own. Now, if you find it's difficult, you're going to make sure when you're back in class, you're going to ask me, and we're going to specifically go over any questions that you have. Please keep in mind you should have an independent practice sheet that you'll do. And again, this is somewhat of a confusing learning target at times, so don't be afraid to go back and make note cards if you need to kind of make sure that you're honing in on, for example, a control is a normal setup. Constants are all the variables that need to be kept the same. The independent variable is the one thing you change and test. And lastly, the dependent variable is what you're measuring and monitoring to see how much difference what you changed and tested made. That's it for now. See you later.